Okay, I'll turn it over to Rory, who is going to introduce today's speaker.
Okay, so it's my great pleasure to welcome Daniel Cadena here to the MVZ. I'm not quite sure when Daniel and I met, but I feel like I've known him forever. It's been a long time. And Daniel really represents one of those scientists that I admire, somebody who's able to both write papers and answer big kind of questions that really integrate lots of kind of data, but still pays attention to detailed taxonomy and those kind of fine in-the-weed things that we care about as well. And so he's been able to really mesh this beautifully with a lot of the work that he's done in the Andes, but also other systems and broader topics as well. So Daniel got his PhD in 2006 from the University of St. Louis, Missouri. And straight after that, he got a faculty position, so he basically left that and started a faculty position at the Universidad dos Andes in Colombia, where he's from, and he's moved through being assistant professor to associate to full professor. And I was just looking on his CV that he was just come off being chair for 4 years, which explains why he's basically on sabbatical at the moment. And I also learned something I didn't know about Daniel, is that he's a keen marathon runner. And he's qualified twice for the Boston Marathon, and will be running it again this year. But I now know why he sort of stayed skinny and remained skinny, and I've kind of gone the other way. So maybe I'll learn something from him. But today he's going to talk about one of my very favorite topics in the world, about tropical mountains and why they're such special places. So thank you very much, Daniel.
Thank you, Rory and Michael and everybody. It's an honor to be here. I was here at an MVZ lunch maybe 10 years ago, and I saw something on hybridization and salamanders being shown here, and I thought, well, this is a very special place, and so it's a real honor to be here. So like Rory said, I've been working on the issue of tropical diversification for a few years now. I've been a faculty at Universidad de los Andes for 10 years, and this is a special place that many of you probably know for very good undergraduate students. So much of the work I'm going to be showing today has been done with Colombian students, many of them undergrads, and I wanted to start by acknowledging that. So on to it. So everybody knows, like, these maps are not news for anybody. Diversity is not homogeneously distributed across the globe. So tropical areas in various regions are renowned for their high tropical diversity. This is a map for vertebrate diversity. If you zoom in to the New World, so diversity is clearly concentrated at tropical latitudes with high species richness both in the lowlands, in the Amazon basin, but also in the Andes, and particularly in foothill areas in the Andes, where you get sort of the combination of lowland and highland faunas. But if you look at a very special component of biodiversity, which is narrow-ranged species, endemics that have small ranges, they are largely concentrated in tropical areas. So foothill areas in western Colombia, western Ecuador, and the eastern slope of the Andes in Colombia, Ecuador, and Peru are renowned for their high concentration of endemics. And well, I come from Colombia, and people sort of brag about us being this mega diverse country, and people sort of feel proud about, well, we have a lot of bird species and we're #1 in bird species in the world. But just to give you a feeling of what that means, I often ask people in Colombia, well, yeah, you brag about us being highly diverse. How many bird species can you name? Like, I would ask my mom, tell me 10 species of Colombian birds, and she'll have a very hard time coming up with 5. But something she did the first time I asked her this, she said, well, there's the hummingbird. But it turns out that if you live in Colombia, the hummingbird means more than 200 different things. This is not looking very well, but anyway. There's something funky with my presentation. Anyway. And just to give you a flavor of what I mean by high tropical diversity, I want to show a few pictures of hummingbirds, all from Colombia, to highlight that it's not only species rich, but it's rich in a bunch of different ways. It's rich in colors, it's rich in behaviors, rich in habitats, life history strategies, breeding strategies. I mean, it's just overwhelming. There's a lot of diversity there, even within clades, like such as the Colombian hummingbirds. And well, none of these pictures were taken by me. They were all taken by a photographer named Murray Cooper. But growing up in Colombia, I sort of became fascinated by the question of, well, why is it that diversity is so high? And of course, I'm not going to be able to answer this question across my career, and this is a topic that has interested naturalists for many years. But I've been working on sort of trying to get at the question of high tropical diversity, and particularly high tropical montane diversity from two perspectives. One is looking at diversification, so looking at some old and newer ideas about the origins of diversity, and that's what I will be focusing on today. And then there's the question of, well, there's all of these species that are being produced, but how do they come together? So sort of in this ecology-evolution interface, how do you get a species coexistence? And in particular, I'm interested in the mechanisms that drive turnover in species assemblages with elevation. How is it that you can pack a lot of species, say, within an elevational gradient? And I'll touch on a little bit about this, but I'll focus mostly on the first issue. So for several years, we've been doing phylogeographic studies, like much of work that has been done here at the MVC with some groups, sort of asking the question of how many lineages are there within species, how are those lineages distributed. Sorry, my slides are not looking very well, but I'll try to make the general points here. So we've been doing studies with probably completed some 10 species or so, where we sample broadly across the neotropical montane region, trying to get all major mountain systems and sampling broadly across taxonomy. So that involves a lot of fieldwork to get specimens, but also getting loans from collaborators and museums in different places. So this is simply an example of one of the species we studied. This was work by one of the undergrads in my lab. And the point I want to make here is that what we're finding consistently across many birds in the Andes is something like this, where populations are highly structured. So within this single species, we found at least 8 or 9 different clades that are highly divergent from each other. And those clades made a lot of sense geographically. So they're typically separated by barriers. So you might find one clade in one mountain, another clade in a different mountain, or within a mountain's range, say the eastern Andes of Colombia, you might have clades on different slopes of the mountain, or the northern and southern sectors of the mountain, which are separated by a dry valley. So this speaks to the role of geographic barriers promoting diversification. And of course, that's not new. We know that species form largely allopatrically. But in complex landscapes such as the Andes, we see a lot of sort of ongoing diversification associated with those barriers. So that's, I would argue, one of the reasons why the Andes are so diverse. Topographic complexity increases the opportunities for allopatric speciation. And an important point I want to make is that not only do we see populations diverging, often at fast rates, but we also see persistence of populations for a very long time. And that's, I think it's an often neglected component of speciation, because you might have things that split off a lot, but if they don't persist, they won't become species in the long run. You need both lineage splitting, but also you need these populations to persist long enough so that they can become different species. And this is just to give you a flavor of the sort of things I'm talking about here. This is work we've been doing for several years with John Click and others, looking at gray-breasted woodwrens. So this is considered a single species, with some exceptions maybe, that ranges broadly from Mexico all the way down to Bolivia in mountain areas. And this is simply a mitochondrial DNA tree that is scaled to reflect time, millions of years before present here. And what I want to highlight here is that within the single species, that is a common bird that you find pretty much in every mountain forest in the neotropics, there's a lot of genetic diversity. There's a lot of structure. So basically what I did here was that I drew lines at different moments in time. And the point here is basically the following. Within this single species, there are at least 6 lineages that are at least 4 million years old, 12 lineages that are at least 3 million years old, 25 lineages that are at least 2 million years old, and 42 different lineages that are at least 1 million years of age, within a single species. Some people might say, well, those are multiple species, probably. But the point here is that they all look alike, they sing alike, and they're isolated in different mountains. So not only are they differentiated in different mountains, but they are persisting for a long period of time. So I would add that it's not only rate of lineage splitting, but it's also persistence of populations over time that might lead to accumulation of diversity over the long term. Then in terms of barriers, we've also been interested in sort of looking at across assemblages, what is the role of barriers in influencing diversification. And I collaborated with a bunch of folks led by people at Louisiana State Museum a few years ago, looking at phylogeography of birds in the lowlands. So this is not the highlands, but our birds occurring in tropical lowland forests, where the Andes is largely considered a barrier that splits populations to the east and west. So what we did in this study was we looked at 27 different lineages of neotropical birds with a bunch of different ecologies, dispersal strategies, diets, etc. And I don't know why my graphs don't look right here, but the sampling was pretty powerful across the neotropical landscape. And what we did was ask the question of whether divergence across those barriers was structured in such a way that different species with different histories and ecologies had common signatures of diversification across barriers. In particular, we wanted to ask the question of whether the origin of barriers was directly related to sort of large-scale vicariance across species. And one of the simple predictions you could make was, like, say, if the uplift of the Andes fragmented populations on both sides, and that led to diversification, then species should show concordant patterns of differentiation across the Andes at the same period in time. And we did sort of the same analyses, not only for the Andes, but other major barriers in the neotropics, such as the Isthmus of Panama here, the Amazon River, the Madeira River, and the Negro River. And what we find is far from a pattern of congruence that you would expect. So these are all the different barriers, the Andes, Isthmus of Panama, the Negro, and the Amazon, and the Madeira, and each of these dots is a species. And here you have the time of divergence across the barriers. So if you take the Andes, say, we have species that clearly diverge across the Andes very recently, within the past few thousand years. But we also have species that diverge across the Andes many million years ago, up to 8 in some cases. And the same is true for all the other barriers we found. We don't see this common pattern of diversification at the same period in time. So based on this and other ideas that we developed in that paper, what we propose is that it's not likely the mechanism that is driving high tropical diversification is likely not landscape change. It's not a mountain uplifting or a river forming or climate changing, such as in the Pleistocene. But it's, once you have a landscape that is already structured, you have mountains, you have rivers, it's dispersal of species across those barriers, which initiates allopatry. So what we find here is that different lineages have, say, dispersed across the Andes at different moments in time. And it's those rare dispersal events what initiates differentiation in allopatry, not the sort of long-thought idea of large-scale vicariance. Okay, so now I'd like to switch to talk about projects we've been working on, looking at sort of large-scale diversification in a few groups, which I think are sort of relevant to understand the questions relating to high tropical diversity. And I've been working on these birds for a few years now, the Tapakules and the genus Cytolopus. So these birds are hard to see. They live in the understory of montane forests. I'd like to say they behave more like mice than like birds. They don't fly very much. They actually don't have fused collarbones. They don't have a wishbone. They have these tiny rounded wings, and they run more than what they fly. And so it seems like it's a group that would be well suited to answer questions related to diversification in a complex landscape, because you would expect they would readily diverge in allopatry and association with barriers and things like that. The taxonomy of these birds has been very poorly known. In the early 1990s, taxonomy recognized only 9 species in the genus. We're up to more than 50 now. We've described 5 of those. Actually, one just got published 10 minutes ago in the OC. I just got a note here from the Andes of Colombia. And we've been working on a phylogeny of these birds for a while now. And here's the reason why these things are so poorly known. I don't know how many bird taxonomists are in the room, but and I won't embarrass you asking you how many species are there here. The answer is 8. And you cannot tell them apart, basically. So they are all very similar. And the reason why we know now they're different species is because they sing different songs, and we know they're genetically different. they typically belong to different clades within a single mountain chain. So actually, these are 8 species that replace each other along mountain gradients in Ecuador. And, well, there's a good reason why traditional taxonomists didn't realize they were multiple species, because they all look alike. So anyway, for the past few years, we've been building a phylogeny of Tapakules in this genus, which is finally complete. I hope to write this up within the next few weeks or months. And we've sampled essentially all named taxa and multiple populations of unnamed taxa. And we have a phylogenetic tree that we can calibrate reasonably. And we've been doing a bunch of things with these groups. There has been a lot of debate in the literature about this software called BAM. I don't know if you've been following. But what I want to do here is simply make a very simple-minded point, which is diversification is very fast in these Tapakules. I won't make claims about changing rates in different clades. We actually find a clade that seems to have accelerated rates of diversification. But relative to other systems in which people have used similar methods to study diversification rates, it turns out that Tapakules have diversified extremely fast, faster than cases like Darwin's finches, and in the same ballpark as things like some of the hummingbird clades that you guys studied in your current biology paper. And of course, I started my talk with showing you 20 different slides of hummingbirds. I didn't show you 20 different slides of Tapakules because people would fall asleep fairly quickly. But the point here is that, well, it seems like even in these cryptic groups, we're having these very rapid radiations. And you could probably call this a non-adaptive radiation because they all look alike, they do pretty much the same, but there are a bunch of different species that have radiated rapidly, probably as a consequence of landscape change and isolation in South America. And if you take the Tapakulo phylogeny and actually zoom out to other genera in the same family, we can build maps like this, in which what I'm showing here is, so the figure is pretty busy, but there are basically 2 things. So you can see there's 2 different colors, blue and green, and there's 2 different shades of colors. So there's bright blue here and say dark blue here, and there's dark green here and bright green here. So blue means early divergent groups, so deep branches in the phylogenetic tree. Green means branches near the tips of the trees. Dull means species poor, and bright means species rich. So say, here in Amazonia, it's species poor because the color is dull, and it's old lineages, it's blue. Whereas here in say, southern Chile and the South Temperate South America, it's species rich, this is bright, but it's also blue. So it's old species, but it's species rich. And if you go up to the north in the Andes of, well, basically northern Peru, Ecuador, and Colombia, what you find is a high concentration of cells with bright green color. So that means it's highly species rich, but it's species that originated fairly recently. So the point here is that these guys probably originated in some areas in southern South America. They colonized the northern Andes, and something happened there, and they just went crazy and diversified very quickly. And this explains why the rates of diversification are high, and it explains why the highest diversity of this group is concentrated actually in areas that were recently colonized, like the northern Andes of Colombia. Okay, so I've talked about rate of differentiation, and also mentioned persistence of populations over time. And actually, in this review in which Rory participated, they highlighted that mountain regions worldwide are hotspots both of richness for recently derived species, but also for old species. So those are areas in white, shown in this figure here. So mountains, to use the jargon that people have used recently, are both cradles of diversity, where recent species are produced at high rates, but they're also museums, where things persist for long periods of time. But the other component of diversity I mentioned is this idea of species turnover. You might have that places on mountains are not very species rich relative to the lowlands in terms of alpha diversity, but beta diversity is very high in mountains. So as you move across the elevational gradient, you get this pattern of replacement of related species, and that leads to the Andes being probably the part of the world where species turnover in birds is highest. And I've been interested in understanding the mechanisms behind species turnover. Many of those are largely, or could be, ecological. So people have talked about competition. We've been doing some work that I can tell you about later if people are interested about maybe competition mediated by pathogens. So we're looking at the effect of parasites on elevational distribution patterns. But of course, there's the idea that speciation could also contribute to stratification of species on mountains. And actually, tropical birds have these very narrow elevational ranges. So here I'm showing data for 3 families of neotropical birds. And what I did here is show the elevational range. So this is elevation here for species in 3 different families, and they're ordered from lowest to highest. And the first point I want to make here is note that none of the species encompasses the full elevational range of mountains in South America. You get relatively narrow elevational ranges with assemblages shifting from the lowlands up to the highlands. And if you take these 3 families and you calculate the median elevational range, so the number of meters over which a species is found, it's interesting. You have to believe me here, my slides are just not coming out right. The median is pretty much the same across the 3 families. It's 1,000 meters. So in areas where mountains might have 4,000 meters or more, the typical species has only about 1,000 meter elevational range. So then the question is, well, why? And like I said, a possibility is speciation. Maybe you have these elevational replacements because you used to have a widespread species that speciated into 2 daughter species. One is in the highlands and one is in the lowlands. And for several years, I've been trying to sort of find examples of this, largely inspired by the work that was done here at the MVZ by Jean Patton on mice in Peru. And one of the places I thought it would be an ideal scenario to look for parapatric speciation along an elevational gradient is the Santa Marta Mountains of Colombia. So these are mountains that are in the coast, in the northern coast of Colombia. It's the tallest coastal mountain on Earth because it goes from sea level up to more than 5,000 meters elevation over a very narrow, very short distance, less than 20 kilometers in some cases. And this is the place we've been working on in Santa Marta. And I'm going to tell you about a study in which I collaborated with Rory a few years ago. So if you look downslope in the mountain, this is what you see. You see typical mid-elevation forest. So this picture is taken at about 1,500 meters. That is humid, but as you go down, it grades into more dry forest and warmer forest. And when you get down here, you're essentially in thorny scrub, and that's the ocean down there. If you turn around the trail and look at the other side, this is what you see. So this is lush montane forest with wax palms that are typical of mid-elevations in Colombia. You have this belt of clouds that come and go every day in the afternoon, especially. It's very densely covered in clouds. And if you're lucky, you can see the snow peaks up in the mountain. We didn't work up here, but basically we worked in this part of the gradient extending down here. And one of the reasons I thought this was an ideal setting to look for parapatric speciation was, one, it's isolated, so things are not going to be getting there multiple times. And then the other thing is that we have the steep gradient. And a third thing is that we had these wood rings, which I just told you about, that they showed some differentiation with elevation that was actually recognized by traditional taxonomists. They were named subspecies from the highlands and the lowlands. So we asked, are these things the product of parapatric speciation, of a single lineage arriving in Santa Marta and diversifying into 2 different species? So the first thing we needed to do was characterize the variation across the gradient, and this is the project we worked on with Rory. So this is elevation here. We sampled a transect encompassing more than 1,000 meters elevation, did about 100 individuals, and we looked at a bunch of things. Microsatellite markers, sequences of nuclear intron, mitochondrial DNA, and we also looked at morphometrics, and we looked at SOMs as well. And what I'm showing here is to simply make the point that basically what you have in all these plots is the probability of assigning each individual to one or another group. So the point is, there are essentially 2 groups of birds here. It's not a transition from one to the other. So if you look at morphometrics, you have all the birds in low elevations are one thing, and then abruptly, all the birds at higher elevations are some other thing. And you get the same pattern with mtDNA, with nuclear introns, and with microsatellites, perhaps with one exception of a bird that seems to be a hybrid, but it's actually located at higher elevations. And an important point here is that the transition between the 2 forms occurs at the same point, regardless of what kind of marker you look at. And the other thing is that there's this very narrow elevational band where the two things are sympatric. So we have adjacent territories. So basically what we concluded is, well, these two things are different species. It seemed then like a perfect case of parapatric speciation along the elevational gradient. But we were missing the crucial piece of evidence of, well, are these things sister to each other? I was pretty sure they were going to be, but it turns out that things don't work out as you expect oftentimes. So we've been working on this phylogeography of this bird that I showed you earlier, from Mexico all the way down to Bolivia. And this is where the two clades of the Santa Marta groups fall. So they're not sister to each other. So it turns out that even these birds with tiny wings that do not disperse, they've dispersed to Santa Marta twice. So it's not a case of parapatric speciation from a single ancestor, but they got there independently. So no, we don't have evidence of parapatric speciation in this case. We've tried to look at this in other groups, and we did a paper years ago in which we basically took data from the literature and looked at the extent to which sister species across various vertebrate groups show overlap in the thermal niches. So the way to interpret these graphs is if you have a high temperature overlap between sister species, it likely means that they're found at similar elevations, because elevation correlates with temperature. Whereas if you see lower overlap or low overlap, it likely means that things are segregated by elevation. And we did this for bats, birds, frogs, herps, lizards, and salamanders. And what we find is that consistently, temperature overlap in tropical species of vertebrates is high, and it's actually higher than in species from the temperate zone. There's one exception to this pattern, and those are actually salamanders, which show the opposite. But across the other groups, we consistently see that there's high thermal overlap between sister species occurring in mountains. So what this means is that vertebrates, I think, in the tropics, and this is the same result that Patton and Smith got in '92, I think, speciation is largely allopatric within a single elevational zones in different mountains. It is not speciation in parapatric across elevational gradients. Maybe one day we're going to find an example, but I think we've looked hard enough, and this doesn't seem to be general, the idea that you could get speciation with elevation. Typically what you get is allopatric speciation within a thermal zone. Okay, so I've been talking about sort of the standard textbook examples of allopatric speciation. No? You get speciation via vicariance, also speciation via dispersal, and you might also get parapatric or ecological speciation across gradients. I didn't talk about sympatric speciation. I haven't worked on this, but I can probably say I don't think this is very common in birds and not in tropical birds. But what I want to do now for the rest of the talk is to talk about maybe other ways in which speciation in the neotropics might occur that are not entirely novel, but I think are new ways in which we're looking at data that people haven't sort of hinted at before. And the first thing I want to talk about is speciation via behavior. And the idea here is we've been interested in understanding the role of migration, movement of animals across space as a driver of diversification. And in particular, we've been interested in understanding the role that changes in migratory behavior might have for speciation. Typically, when people think about speciation in allopatry, you're thinking about changes in the landscape that, say, fragment populations, and organisms are sort of passive in that role. They just get isolated. Other cases, you might get a more active role, so things disperse across barriers. But here, we're thinking about a mechanism where you don't need to invoke the creation of any barriers. So organisms sort of generate their own allopatric distributions via changes in behavior. So consider a species that is widely distributed, and it's migratory. It breeds here in the temperate zone. It goes to the tropics during the winter, and then comes back, and does that cycle. Imagine that at a given point in time, some individuals stay in the tropics and breed there. And this, for some reason, becomes heritable, becomes established, and you get then these two different populations, one of which is migratory, and the other one is resident in the tropics. they come into contact at a given point in time, so they're not allopatric entirely, but they come into contact at a period of time where one of them is not breeding. So this is a scenario that might trigger speciation because these guys essentially become temporally and also physically isolated in terms of their reproduction. And this, of course, is not an idea of mine. There's a guy called Kevin Winker who has written a bunch of papers on this. He actually wrote a paper calling this heteropatric speciation, in which changes in migratory behavior are what trigger diversification. So to look at this, we've been doing studies at a microevolutionary level, and I'll show you also a study at a macroevolutionary level. So at the micro level, many species of neotropical birds are partial migrants, meaning that some populations are migratory and some populations are resident. And we wanted to ask the question, just to start with, are resident and migratory populations genetically different? And we started off working with a fork-tailed flycatcher, Tyrannus salana from South America, which actually it's an austral migrant. So some populations breed in Argentina and spend the winter up north in the llanos of Colombia. And other populations, particularly from northern Colombia, are resident. They stay at their breeding grounds year-round. So we sampled the species broadly across this range, and we did GBS analysis using SNP data. And when you do a typical structure analysis, what you get is that there are essentially the most likely number of groups in this data set is 2. And those groups separate migratory and sedentary populations. So they actually are genetically differentiated. But if you run these analyses with different numbers of clusters, you actually start to see that within the migratory groups, there seems to be a lot more genetic diversity than within the sedentary population. So the hypothesis here is that maybe the sedentary group is derived from the migratory group. And the low genetic diversity might be a consequence of bottlenecks associated with individuals staying in the tropics. So to test that, of course, you need a tree. And if you build a tree, this is what you get. If you can see it. Maybe this will do the trick. No. Anyway, what I'm trying to show here is that all these deep branches here correspond to migratory populations, whereas the resident populations, the sedentary populations, are up here in this single clade that is clearly nested within a group of migratory populations. So what we think happened here is that actually these birds lost migration. The resident populations differentiated from originally migratory populations. So maybe loss of migration might trigger diversification if these things eventually become reproductively isolated from their ancestors. But to get at this, you actually need to look at the macroevolutionary patterns. And to do that, we built a phylogeny of a whole family of flycatchers. So this is based on published data, but we basically put together all the sequence data out there and generated this tree in which we mapped migratory behavior, separating migrants from partial migrants and sedentary species. And it turns out that the ancestor of flycatchers most likely was sedentary, and we get various instances in black of the evolution of migration, and also, of course, the evolution of partial migration. So one of the things we did here was to look at the sequence of transitions between these traits. There's this hypothesis in the literature that partial migration is an intermediate step in the evolution of full migration. It's called the stepping stones hypothesis. So we wanted to see whether partial migration was an intermediate step or not. And then we wanted to ask the question of whether migrant lineages actually diversify at a faster rate than resident lineages. So basically what my student Valentina Gomez did was look at different models specifying transitions between being migratory, being partial migrant, and being sedentary, and looking at support for different evolutionary models for transitions among those character states. And what she found was the support was overwhelming for this model here, that is expanded here, in which, yes, partially migratory seems to be an intermediate step between migration and sedentariness. But it actually goes the other way around. The stepping stones hypothesis proposes that migratory lineages become, resident lineages become partial migrants, and partial migrants become full migrants. But what she found is that the more common pattern is that migratory lineages become partially migratory, and those partially migratory become sedentary. So the pattern at the macro scale is the same pattern we're seeing within our study species. So things, sure, migration has been gained, but it has been lost a lot more often than the rate at which it has been gained. So then we ask the question, are migratory lineages diversifying at a faster rate relative to resident lineages? And the answer is overwhelmingly yes. So this is estimates of diversification rates across a bunch of different phylogenetic trees. These are resident species here, these resident lineages here, these are migrants, separated into migratory and partially migratory. And it seems like rates of diversification in flycatchers are actually higher in migratory lineages. So this hypothesis that changes in migratory behavior might trigger diversification seems to have some support. Okay, and the final thing I'm going to tell you about is this other idea about speciation mediated by isolation in time. This idea of allochronic speciation. So that, of course, is not new for people who work on insects. So there are excellent examples in cicadas and crickets and even salmon, showing that populations can breed at different times of year, and breeding at different times of year causes reproductive isolation because individuals are not meeting for mating. Well, it turns out that birds are highly seasonal in how they breed, even tropical birds. So this is data for spotted antbirds in Panama, or from Werkelski and his colleagues. And what I'm showing here is, these are males here, there's some females here, but we can take the male data. So these are testes sizes. So what you can see here is that early in the year, males have small testes, so the gonads are not active. And then gonads start growing and become active at a given point in the year, and then regress and decline in size. And this cycling pattern, which is repeated across years, is related to precipitation. So it turns out that during the dry season, birds are not breeding, and their gonads are small. But at some point during the dry season, they perceive some sort of cue in the environment, and their gonads start growing. And they start growing to the point that when they start breeding, it's pretty much at the beginning of the rainy season. And that guarantees that when they're raising young, there's going to be a lot of fresh foliage and insects. So birds seem to time their reproduction to coincide with peaks in resource abundance that are likely mediated by precipitation. So I think this shows that reproduction in birds is highly seasonal and it's related to rainfall. And then the other thing is that, of course, rainfall is variable. It's variable in time and it's variable in space. So this figure here shows for a given moment in time a map of the world showing places in purple are places where it's raining a lot. Places in red are places that are dry. So if you look at here, say it's very dry in the Amazon basin. It's very humid in the Amazon basin, sorry. South of the Amazon River, but it's dry up north. And I'm going to circle through a full year. So this is January. And if you focus your attention here, you'll see what happens. It's February, March, April, June, May, June, July. So see what happens in July? The pattern is totally inverted. It's now dry here in the south, and it's wet in the north. And you can pick other places in the globe to look at this, and you will see that you get these sort of changes in rainfall regimes. So the idea here is that if avian reproduction is seasonal, and it's linked to rainfall, and there are seasonal patterns in precipitation that vary in space, you put those two things together, and you might get a pattern where birds might breed at different times in different places. And actually, there's nice examples of this. This is a beautiful work done by Ignacio Mora and his colleagues in Ecuador. So these are populations of rufous-colored sparrows that are separated by only 25 kilometers in localities in the Andes. Yet these localities differ totally in their precipitation regimes. So say here, in the month of June and July, the white locality is experiencing high rainfall, the black locality very low rainfall, and then later in the year the pattern is inverted. And when you look at the breeding condition of birds, so these are levels of testosterone circulating in blood, you can see that, say here, the black population, birds are reproducing, testosterone levels are high, but then a few months later, those testosterone levels are very low, and then here it was low, and it becomes high. So populations separated by 25 kilometers have different breeding schedules. So the question is, can this promote differentiation? And I had been thinking about this for a few years actually, but I didn't have any data to sort of test it and didn't know how to test it. And when I was still scratching my head about how you might go about testing it, I came across a paper published in a journal that I didn't know existed called Ideas in Ecology and Evolution. So it turns out you could publish an idea without having any data. No, but full credit here to Paul Martin and his colleagues who actually wrote this down. And they proposed this, what they call the asynchrony of seasons hypothesis, which basically goes like, if you have spatial asynchrony of seasons in precipitation among localities, specific populations might have different phenologies. Those asynchronous phenologies might limit the establishment of individuals, and that might reduce gene flow and effective gene flow among populations, and that would lead to population divergence. So what we thought, well, now that there's this framework, we could maybe test this hypothesis. And the idea would be that if you have different localities, say here in the map, and these different precipitation regimes, you could somehow calculate the asynchrony in rainfall among localities and relate that to, say, genetic distance among populations. And the prediction would be that populations that are more asynchronous in rainfall would have more genetically, would be more divergent genetically, assuming rainfall mediates reproduction. So we did that study with my student Ignacio Quintero, who was an undergrad back then. He's now a PhD student at Yale. And basically, the idea was like, if you have, say, this population here and this population there, where this is a peak in rainfall, in the pattern in rainfall in one population, and this is the other one, you can see here that asynchrony is complete. When one population is at its peak in precipitation, the other one is at its lowest valley, and the other way around. Whereas these two populations here, they have fairly synchronous, these two here have fairly synchronous precipitation regimes. The displacement between seasons is only 15%. So the idea here is that we would expect a lot more gene flow among between these populations than between these populations here. So I'm going to move fast here, but basically what we did was we collected a bunch of data from the literature for studies that had mitochondrial DNA phylogeographies and good locality data. We calculated asynchrony in rainfall among localities. I can tell you how we did that later on if you're interested, using Fourier spectral analyses. Using high-resolution climatic data from essentially radar data for clouds cover that gives us very fine resolution about spatial patterns in precipitation. And then what we have is a matrix showing asynchrony in precipitation and a matrix showing genetic distances among populations. We can relate those two. Of course, it's not that simple because I spent like 20 minutes talking to you about the role of barriers, so we cannot ignore the role of barriers. But we sort of consider the role of barriers by generating ecological niche models for all the species. And based on those models, we could calculate resistance distances that sort of indicate how likely is it that species might disperse among localities based on the climate. So basically what this allows us is to take away the effect of barriers and see the contribution of asynchrony in precipitation to genetic distances. And we did this for a bunch of neotropical bird species, close to 60, and this is what we find. So this here are coefficients of a Mantel partial analysis, where it's kind of a regression analysis where you look at the association between asynchrony in rainfall and genetic distances. So 0 would mean no association. Positive values would indicate the predicted relationship, positive by the hypothesis. And what we find is that, well, there are a lot of species that do show a positive association. So populations that live in more asynchronous environments are more genetically divergent. And we calculated a cross-species effect using phylogenetic meta-analyses. And it turns out that the cross-species estimate is positive, and it's significantly different from 0. So we do see a pattern where more asynchronous populations are more genetically divergent. Well, this is very interesting. I was very surprised by this pattern. But I haven't told you where's the link with breeding seasonality. I'm making a lot of assumptions here. I'm saying asynchrony in rainfall means asynchronous breeding. But I didn't show you any breeding data. We've been trying to get at breeding data, looking at specimens. And the point I want to make here is that this is very hard to do. So what I did was we took the most commonly collected species in bird collections worldwide for neotropical birds, which is the wedge-billed wood creeper. There's a lot of specimens with good gonad data, so we wanted to see whether populations from different sites actually breed at different times. And it's pretty sad because there's a lot of data in terms of specimens, but when you put that on a map and you sort of separate it by times of year, and blue means birds in breeding condition, red means not breeding, there's not a whole lot you can say. First, the data are very scattered in space, and they're very limited. Maybe you can see here with some imagination that things are breeding more here, like south of the equator, and they're not north of the equator, but I wouldn't do statistics on that, you know. And the point here is that we really need more data, and simply the most commonly collected species of neotropical birds is insufficient to do an analysis of this sort. Even for those species, we need more specimens to get at these sorts of questions. Let's get this. Okay, so I've talked about birds, and I've talked about this assumption that rainfall relates to breeding, but I'm making a lot of assumptions. So the next thing I thought, well, let's look at a group in which water availability is more directly tied to reproduction. And there, well, we found a strong effect in birds. If this is general, we're going to find a very strong effect in things such as amphibians. So we looked at, this is work in progress, we did this analysis last week actually. So we looked at 60 species of neotropical frogs, and we repeated essentially the same thing I showed you for birds a minute ago. And what we found was a total bummer for me, actually. Because it turns out, well, I can make a graph that looks sort of similar to the one I showed before. So values of above 0 indicate positive effects of asynchrony in rainfall on genetic divergence. But I mean, the median is essentially 0. So there's no effect whatsoever of asynchrony and rainfall on genetic differentiation among populations of frog species in the neotropics. So at first I thought, well, maybe we did something wrong with the birds, we need to reanalyze the data. No, the signal is there, we use the same methods and it's there. But then I got a little bit philosophical and I started thinking, well, maybe there's an artifact of taxonomy here. And I don't know about this, there are a bunch of herpetologists in the room, but I have the feeling that people that work on frogs of late have very little tolerance for within species variation. When you find things that are different, you split them. At least that's happening in South America. I don't know about here. That's true. So maybe you're not seeing this in amphibians because anytime that there is genetic structure, people have split species. So then what I said, well, then we need to look at this at a higher level. We need to take sister species of amphibians and see how asynchronous they are in terms of rainfall. And if this has been important for diversification, then sister species of amphibians should be more asynchronous in their environments than non-sister species, or something like that. So based on that, we took the same data, well, we needed to find more data because now we needed pairs of sister species, and we were only able to come up with 10 of those, for which we had enough data. So you can estimate asynchrony in precipitation for these 10 pairs and calculate a distribution for that. And sure, you get a number, but then the question is, how does that number relate to a null distribution of things that are not sister to each other, so certainly did not speciate according to this mechanism. So I showed you the figure before where the median effect was 0. This is the null distribution for 100 species of amphibians that are not sister to each other is indistinguishable from the distribution of sister species. So I think I have to say this doesn't apply to amphibians apparently. And why it would apply to birds and not to amphibians is something I don't know. I don't want to defend my hypothesis very much, but a possibility is that, well, maybe amphibians are not that hardwired after all. So if they disperse to a site where it's raining, they'll breed. Whereas birds cannot really do that because they need, well, part of their reproductive schedule is herdable, but also it is plastic, but plasticity requires time for adjustment. So if they get there at the wrong time, they still would need months to have their gonads grow, and they're not going to be ready to breed. That's sort of my explanation to defend this, but I'm happy to say this might not be general. And what I'm trying to say here is that while these hypotheses might be relevant to understand patterns globally in terms of species richness, so actually Martin's paper was published in a very specific context. And he said that this asynchrony of seasons hypothesis would largely apply to tropical organisms and not to organisms in the temperate zone. Because in the temperate zone, to a large extent, things are not synchronizing their reproduction in relation to rainfall. They're doing it in relation to temperature and the length of the day. So birds breed in the spring, they're not going to breed in the winter for the most part, regardless of how much is it raining in the winter. And I don't know, I think it's just intriguing that these patterns might be partly explained by explanations such as that. And like Meyer said more than 50 years ago, I think we know very little about the role of asynchronous breeding in reproductive isolation. And what I like to simply say is that I think this is a hypothesis that needs more attention from us. So to finish up, I basically showed you the two examples I talked about, the speciation mediated by changes in behavior in relation to migration, and speciation associated with asynchronous breeding seasons, I think are unique in a particular way in that we've typically been thinking about tropical diversification, invoking sort of large changes to the landscape, you know, like mountains uplift, rivers form, climate changes and fragments populations. Sure, that's probably very important. But also the point is that given the complexity of the tropical landscape, once it's there are a lot of mechanisms that could lead to diversification that do not require any further changes in the landscape. A structured tropical landscape allows for speciation via dispersal across barriers, but might also allow for situations like those I showed involving speciation via changes in behavior or changes in breeding schedules. And with that, I'll simply end by emphasizing that people have been interested in diversity in the tropics and mountain systems in particular a very long time ago. We were just talking about this beautiful figure from Humboldt, 1804, looking at replacement of related species of plants in the Andes of Ecuador, the decline in species diversity with elevation. And of course, all the questions and issues I talked about today are not new. But I guess the point is that when you look at those issues from the lens of history and phylogeny and evolution, there's a lot more to learn than based on the sort of the static view of patterns being explained by ecology. And with that, I'll end. And I simply wanted to note once again that all of the work I showed here has involved, well, many collaborators, including Rory here and other people, but also students from Universidad Los Andes, master's students mostly, but also a bunch of excellent undergrads. So pay attention to students from Los Andes that want to come here, because they're very good. And I realized this yesterday that I think I'm getting kind of old because I already have like 4 generations of people in my lab, so 4 pictures of totally non-overlapping generations. So it's been 10 years and that's what happens. Time so fast. But anyway, it's been a pleasure to be here. Thanks for listening. I hope this was of some interest to you.
People who need to escape their classes get out, but we certainly have time for questions.
Yes.
All right, questions?
Yes.
Thinking about the results of frogs versus birds and the difference there. I was wondering maybe if for frogs, the landscape resistance explains so much of the variation.
There's
There's
very
not
little
much
to
left
explain.
over because of the different dispersal.
Agreed. Yeah. Yeah. Actually, I didn't show you that. So we did like a multiple regression kind of thing with frogs, and of course, like topography explains a lot. So there's very little left to explain. But even that you cannot explain based on seasonality. Yeah. Right.
So I was quite intrigued with the migratory idea, and so I've always thought that with the migration, it's pretty dynamic. So if you take migratory species and you retrace like where the distributions were during the last glacial maximum, right? Mm-hmm. And then you can basically demonstrate that they must have, many of them probably became sedentary.
Right.
and then essentially they re-expanded another migration, and then became sedentary again. Do you have enough data in that tree to be able to slice it a little bit about when you get the migration kicking in and seeing how that would play a role? So
Yeah,
like if
we
you
could do
became a
that, because the other thing is, like I emphasize that it's a lot more common to have populations losing migration than gaining it. And there are a bunch of these contemporary examples where populations now are known to be losing migration. Like say we have blue-winged teal breeding in Bogota, Colombia right now, and they should, And a possible explanation for that is that, well, it's now warm and nice, so there's no selection for sort of.
And that would be my argument as climate changes, that selection to re-evolve that.
Exactly. So you might find that in particularly harsh periods in the temperate zone, so the pressure to evolve migration would be stronger than in interglacials that are warmer. And
So it
and
almost
things would
links
settle
your
down.
seasonality hypothesis with your migratory hypothesis.
Well, I think they're linked in the sense that we don't really know a whole lot about when the birds breed, but essentially, if you take the case of, we don't know when birds in the flycatcher we studied, we don't really know when birds breed. But we know for sure that birds that come from the south, they're not migrating, they're not breeding in the middle of the year, that's the winter for them. And anecdotally, we found one nest of the local birds at that period of time. So they might be, so there's this component of geography, but it might also be a component of time. So things not only breed in different places, but they breed at different times. So the migratory birds come to Colombia after the breeding seasons in Argentina. And they're probably not primed to be interested in reproducing, even if the local birds are reproducing. But I guess it might happen in the long run. But I don't know if that answers your question. Yes?
As an ornithologist, what is your thoughts on the concept of species that amphibian biologists have? Or mammalogists, I guess.
Well, I don't really know whether what I said is true or not. I don't know if you guys agree, but
Jim was laughing.
Well, I just know you raised that, and I just thought this paper by George Barraclough et al. That's going to triple the number of bird species
Well, of course,
on the basis of
that's
molecular clay. Are you going to?
well, that's something I don't
Yeah.
agree with.
Well,
to start with.
but I just you may lose your signal.
Yeah.
Once that happens.
Yeah, the thing is, I mean, I don't know as much as the difference of concept, but I'm intrigued by how people are applying species definitions in different ways in different places and in different clades. So I'm a member of the South American Checklist Committee of the AOU. And we are this ultra-conservative club that we won't split anything unless there is evidence that they're reproductively isolated. And that, of course, is super problematic for allopatric populations. We recognize that, but we're conservative. But then there's these people saying that, well, we need to split up everything. And we're getting to this point where I think that avian taxonomy, which seemed to be super stable and super clear, is going to be total chaos. Because people are doing different things in different places. So say BirdLife International just jumped into the bandwagon that they split everything up.
20,000.
That makes sense from a conservation standpoint, but you don't want to have conservation pulling the horse of taxonomy. Then we have people like Barraclough and Zink proposing everything should be split. Then within South America, like say Brazilians, they have this it's a version, I guess, of the phylogenetic species concept that I call the Brazilian species concept. That they build mtDNA trees and they find structure and that correlates somehow with plumage. And this is not a fair treatment, but they've done a lot of work with nuclear genes and stuff like that, but they're not focused on the criterion of reproductive isolation because they say you cannot test it for allopatric populations, but they're splitting things apart a lot. And there's a Brazilian records committee, and the Brazilian list is just growing. We're not doing the same in Colombia. So very soon, Brazil is going to have more birds than Colombia because taxonomists are doing different things. And if you go on to, so I don't know what mammalogists or herpetologists do, but if we're doing different things in different groups, I think it's complicated, especially if your species concept influences what you think is important for the study of speciation and conservation and diversity and everything. you.
So I have a comment about that, but I also have a question that relates to your bird species. I mean, I think historically, I thought that ornithologists were much more likely to split things than were herpetologists, and that might be
Historically, yeah.
changing. That might be changing now, and I have no idea how it applies to the South American lineages. But I have a question about your flycatcher example, where you were talking about the sedentary forms and the migratory ones, and asking whether or not these things might diversify in the future. It seemed like you had some pretty good evidence from your structure analysis of a strong barrier to gene flow between these lineages already. And so what would convince you that you're looking at two species there? What qualifies as evidence of lack of gene flow that is,
Yeah.
I guess...
No, I think this is pretty good evidence. It'd be nice to have some data on the breeding schedules and that kind of thing, but I mean, they're sympatric through part of the year, and they don't reproduce because they're asynchronous. So I would be happy calling them species.
So I had a question about that too. In that phylogeny, the ancestral state was sedentary, right? Was not
For the whole family.
For the whole family.
Yeah.
And the model you presented was if you have a migratory form and some decide to stay put, that could lead to reproductive isolation. But it could go the other way as
Right.
well. Are there any examples where you see a migratory form nested within, a sedentary or a migratory
Well,
population?
there are a few examples. Well, they were in that model I showed you with the arrows, but it's rare. But this is all very labile, and I think it's pretty interesting. There's a beautiful example with swallows, barn swallows. So they breed here in the temperate zone, in the north temperate zone, and they used to migrate down to South America to spend the winters in Argentina. And a bunch of people found that they stayed in Argentina, and they established a sedentary population there. So they lost migration. Then a few years later, they started seeing that those same birds sort of disappeared seasonally. So they thought, well, they started migrating again. But then they fitted them with geolocators, and it turns out that they became austral migrants. So it turns out they now breed in Argentina, and they go and migrate to Colombia to spend the winter there and come back. So they were boreal migrants that lost migration and then gained austral migration within 20 years maybe. So it's pretty dynamic, and it's pretty interesting because if you think of migration being this suite of adaptations, so you need to be able to feel, to navigate a bunch of things. How is it that it gets lost and gained so easily? So it might be sort of a suite of genes that go together. Yeah. People say that birds might have like this common genetic program that enables them to become migratory, and that when conditions are good for it, it can just turn on. But yeah, I would call them species in that case.
All right. Well, thank you very much.
Well, thank you.